today we are doing acquired humoral immunity, which is distinct from the innate immunity that we did last week. Last week, innate immunity was all about the ways in which you are able to deal with viral infection or bacterial infection or any kind of cellular damage that you are born with. This is not something that your cells have to be educated to be able to deal with. This is not something that arises later in life. This is something that is absolutely intuitive to you as a human being. And any other organism that has an immune system, which goes all the way down to bacteria if we're talking about CRISPR-Cas, this innate system is repeated over and over again that is tuned to the things that are dangerous to your kind. Adaptive immunity is more nuanced.
Yeah, so adaptive is this process by which you learn a particular pathogen, your body learns it, and you develop memory for it. And there's two main components that are two main cellular components in that, which are the T and B cells that I mentioned. Together, T and B cells are called lymphocytes. So that's worth mentioning. They have a different lineage than the rest of our cells, and can we actually go back to that chart that shows the lineage of everybody from last time? Way back at the beginning, I think. Yeah, there we go. Yeah. Okay, so as you see over here, on the right-hand side, we see the innate immune system that Dr. Benbury was just talking about. And on the left-hand side, we see the lymphoid system, which is what we're going to be talking about. It has a lot of different names, and I know that's really confusing, but anytime you see the word acquired, humoral, lymphoid, sometimes the word cellular is associated with it, like the T cells that are going out and doing their killing. We're always talking about this side of the screen, which is the T's and B's. And they have, most of these other cells, they sort of evolve naturally. They do a job and then they die. The cells on the left-hand side, these lymphoid cells, they have to become a specific, they have such specialized work that they need to actually go out and encounter the world, that is like meet the pathogen before they end up at the bottom of this screen and are able to actually do a job. So anything that hasn't reached that terminal stage is essentially referred to as naive. It doesn't know what's up. It hasn't been out in the world. It doesn't know what it's doing. So you want to go back down. So if we go to, we're going to talk about the B cells first, and we're going to move over to the T cells. The B cells are all focused around the production of antibodies, which are an extremely important piece of this COVID discussion. Obviously, you hear people talk about, oh, do I have antibodies? This is more or less a surrogate for immunity. Do you have some level of experience and ability to remove this viral system from your body? And so in order to develop this kind of specificity, especially with the antibodies, but honestly, this is true to some extent of the T cells too, you have to hone a specificity. And so I believe, maybe move this into the center a little bit so you can see. We're talking about the generation of specificity here.
Right, so you have a progenitor cell that gives rise to a large number of lymphocytes. And this large number of lymphocytes, each one of these is specific, which you can see here is demonstrated by the fact that they have these very cartoonishly adorable shapes down at the bottom. And what happens is that each one of these lymphocytes are capable of recognizing a specific pathogen that they have been matured into seeing. And then once it sees that pathogen, it is then activated to reproduce massively. So you have vast quantities of these cells that are specific to the pathogen that has been encountered, and then these effector cells are able to eliminate the pathogen.
So it basically multiplies is the very last step. The one that's chosen is multiplied. And honestly, if you just take a second to think about how crazy this is, when your body encounters a new virus for the first time, it goes through this process where it has to seek out the perfect cell to do that job. There might be millions of B cells just hanging out in your bloodstream, and it has to find the one that is capable of best binding to this particular little virion shell, in this case the spike protein, and then it has to get that guy into full swing action. It's got to train him up and get him ready out to go out and multiply and fight this infection. So this is a really, really kind of miraculous process that the body is able to seek out this 1 in a million shot and make it happen.
Except for the fact that it is not magical. It's miraculous, but not magical, right? Because there is a very physical, very straightforward mechanism by which these cells are capable of doing this, and this is the surface immunoglobulin receptor, which is also known as the antibody. And this is for B cells. So B cells have this B cell receptor, which is the surface immunoglobulin, or it can be a secreted immunoglobulin, in which case it is called, it's just this antibody that is not anchored into the membrane. T cells have a T cell receptor, which has a slightly different structure to it, but has the same spirit attached to it, which is the fact that it has a massively broad specificity, very carefully tuned to not have any specificity for the self. So it's this searchlight that looks out into the world.
Yeah, they're capable of recognizing a broad swath of different antigens, but each cell only recognizes one. That's what's really crazy about it.
And so you have basically a population inside of your body of lymphoid cells that are capable of, in theory, of recognizing all shapes that do not belong to you.
And that's what this image, I think, is trying to show, is that in the first step, you have all these different cells, but you're only going to really use one of them
for any given moment. So basically, you have two portions of the antibody, which is you have a variable region, which is where you have this specificity that emerges. And so the tips of these are very, very variable. And we'll talk later about how it is that you have this very specific site that changes from cell to cell. There's a very specific genetic program that is involved in producing this. And
we'll get into that today.
And the constant regions are, depending on what kind of receptor we are talking about, whether it is secreted or it is a transmembrane or it's a T cell, you have slightly different organizations of the constant regions. And then sometimes they're anchored into the membrane, sometimes they are not. So we talk about
and in the case of antibodies, you have other cells that are capable of grabbing onto the bottoms of them, right? So that when you say coat a virus in antibodies, the other cells that come and eat the virus have something to hold on to down here.
And which is illustrated in this next image. Which is basically that when you have multiple different kinds of antibodies that are specific to different things, they're capable of sequestering them in different ways. So the two key words here are neutralization and opsinization. The process of neutralization is if you have something inside of the body that is damaging, antibodies are capable of basically sequestering it. So they're able to bind to the toxin, they're able to create a complex that is large enough and recognizable enough for another cell, like a phagocytic cell, to grab onto and to ingest and to digest, thereby getting rid of the toxin. It's basically like mopping up when there's something bad that's in the body. And then you have another option, which is that you have opsinization. And opsinization is the process by which a foreign molecule is coded for digestion. So it's similar to neutralization in the fact that it is basically just taking something that shouldn't be there and coding it, but it has a slightly different name if you're thinking about neutralizing something that's an active chemical versus coding something that shouldn't be there and then digesting that, like a full virion or a bacterium. And we see that complement gets involved here as well. So basically there's two forms of opsonization. The first form is that when the bacterium or the virion gets coated in antibodies, the constant regions of the antibodies are able to be recognized by the same phagocytic cell. They take it in, they break it down, or it's possible that complement gets involved, and complement is also able to coat the outside of the same structure and help it to be pulled into the right kind of phagocyte.
Complement is something we could have touched on a bit last time, but it is in some sense part of the innate system, where you have this molecule that gets activated immediately, and it does all of these different processes all throughout the body. It's going to activate liver enzymes. It's going to basically gear up. It's one of those sensor molecules that goes off right at the beginning of the infection and sort of puts all of the other systems on alert. This is just another case of that. And it actually does play into COVID. I don't know if we'll come across it specifically or not, but we'll get into it in detail if we need to.
If you've ever heard of cytokines, so there was a brief moment at the very beginning of the pandemic where there were people that were getting really, really, really sick and dying in huge numbers in Spain and Italy. It was before that it had sort of come to the United States and the science had started to sort itself out. A lot of people were talking about cytokine storms because the Spanish flu in 1918 is thought to have been so deadly because it generated a cytokine storm. And part of that has to do with activating the complement cascade, which then basically kicks the immune system into insane gear, and you get whole body inflammation to a degree that basically liquefies all of the borders between your cells. All of your structures are unable to maintain stability and integrity, and then it's just this feedback loop, which the end is basically sepsis and then death.
And complement's also involved in the clotting cascade too. It's a big part in activating some of those thrombogens. So that could also play into some of the adverse effects that we're seeing with this too. So we'll get into that in the literature though. All right, so so where's all this happening? This is a bit strange, but you have a completely separate ductile system in your body responsible for the lymph circulation. So it's generally speaking, your lymphocytes, the ultimate producers of these antibodies, and really the soldiers that are going out and removing specific pathogens, specific antigen tagged, actors from your body. They're just walking around the body. Like I said last time, like this is a very unique cellular system in the body. It almost resembles an amoeboid. These things move around on their own. They kind of walk as they go. They can squeeze through blood vessels. But once they've been activated, right? Once something has been seen in the body, they need to go back to their little institutional home and get to work on cranking out a remedy and educating that 1 in a million B cell and get that B cell to work, or that 1 in a million T cell and get that specific T cell effector to work. And so the highway that they use to do that is this lymph system, the lymphatics and the lymphoid tissues. And so I think what we want to get across here is just that much of the precursors, the naive, uneducated cells, are being developed in the bone marrow. However, and making their way either to some secondary, they're making their way to a secondary organ. For instance, like
the T cells go to the thymus, so they leave. So there's the naive or the immature cells, and then the fully mature cells. So both are produced in the bone marrow, both B and T cells. And what happens for the B cells is that they remain inside of the bone marrow, and that is where they undergo maturation, which means that they are exposed to antigen that is foreign, they're exposed to antigen that is self, they become cells that have matured and have been already selected for to make sure that they're not going to attack the body itself. T cells, however, will leave the bone marrow, and they will go through the lymphatic system to the thymus, where their education is completed. And so an easy mnemonic to be able to memorize this is B cells in bone marrow, T cells in thymus.
Those are the primary organs for those. So they will have to go, eventually the B cells especially are going to go and hang out with T cells in the secondary lymph organs like the lymph nodes and the spleen also. And they're going to do a lot of their maturation there, which we'll get into the nitty-gritties of in a second.
One thing that I really want to point out about this diagram is that up until, I think, 2013 or 2014, it was not clear whether or not there was a connection between the lymphatic system and the brain. And it was just very, very recently that the actual ductile systems inside the brain that carried the lymph fluid and the immune cells were discovered. And so up until that point, it was understood that there must have been some kind of immune surveillance in the brain, but no one had been able to identify the places where it was actually occurring. So if you look at, for example, you have these pairs patches in the intestines. These are very, very clear structures that if you were to take a scan of the intestine, or if you were to do a colonoscopy, you can actually see these patches on the surface of the intestines. And these are basically little agglomerations of secondary lymph organ where immune cells are basically encountering the environment. And there wasn't a similar understanding for the brain until very recently. And now we know. The lymph system circulates separately from the blood to some degree, but then it will rejoin the blood and then will be filtered back out to go into the blood. So these things are basically constantly mixing.
I was going to say, yeah, it depends essentially on you moving around, which is, I think, probably pure speculation, but I think that's a large reason why exercise is so important in fighting infections. You can't actually move your lymph just by sitting in a chair, at least not very effectively. So things like even breathing, like just like really heavy breathing is enough to move your lymph around. And so until it's rejoined the blood, it's just going to sit there and it's going to be hard to do its job. So.
Yeah, it doesn't have a pumping organ the same way that the blood does, right? So when the blood pumps, you have the heart that creates pressure, but the lymph doesn't have the same process. Like it gets a little bit of secondary pressure from the blood pressure, but honestly, your muscles are the things that are pushing things back. And if you encounter a pathogen at the periphery, you have to be able to get that cell back into one of these organs where it's able to proliferate in order for it to be able to actually alert the body that something has happened. So if it remains at the periphery, it's entirely possible for the infection to spread without the alarm bells to precede it.
Same, just a different picture of the same thing, but we're basically talking about the drainage system. Moreover, similar. Anything to add here?
This is something that you can kind of tell inside of yourself, right? So if you've ever had a sore throat, you can reach back to these lymph nodes that are underneath your jaw. And if you have some kind of, if you basically have an upper respiratory infection, you will feel these harden.
The doctors always do that to you when you go in.
Yeah, they always test you. I will oftentimes, if I have, like I get pimples, and if I have like a big pimple on my chin, the lymph node right here will react. And so you can basically start to feel this on your own body. And so what does it mean for a lymph node to get big like that?
Well, yeah, exactly. So as soon as some you can see in this picture here, there's a cut this gentleman seems to have down on his ankle. And what's going to happen is that the sentinel cells there, whatever they are, we talked about all these innate actors that are capable of sending out messages to the other cells. They're going to work their way to the nearest lymph node through the lymphatic system there. And at the lymph node, they're going to start sending out calls for more folks to show up and help them out. And so, along with all these generalized inflammatory actors, you're going to see an influx of cells into the lymph nodes as they gear up to do their adaptive jobs, which we're about to get into. So here is a lymph node. How much of this, we might want to wait until we talk about actual maturation for this to make sense. But you can get acquainted with it. There's different compartments here. What's important is that there's a place for each cell type inside of the lymph node, and the same could be said about the spleen. It's going to be a place where the T cells come and teach the B cells how to basically make antibodies, and we're going to go through that. I think we got a video for that. And there's a place for the B cells to come in, and there's different regions, and you can actually see this happening inside of the lymph node itself, so you can see what stage of infection somebody is in by, sometimes they'll biopsy these lymph nodes to look at this.
And I think that what's important here is to see and to note the organization, because what you have is on the outside wall of the lymph node, you have these afferent lymphatic vessels, which means that the cells, these sentinel cells that are in the tissue, are coming into the lymph node from the back of it. And the back of it forces these cells to cross across the lymphoid follicle, which is mostly B cells. And so they encounter the B cells, then they encounter the T cells, and then after that you have the drainage of the lymph node. And so basically,
this is where all the antibodies are being cranked out too. So this is a huge factory once it's geared up into action. So you need to drain all those antibodies into the blood too.
I mean, honestly, I look at this as almost like a proto-kidney, where it's like there's filtration that's happening across it that is forcing these cells to come into contact with something that has come from the periphery in order to generate the molecules that are necessary if something has arrived with a signal that something is wrong. Because you cannot get back out through the lymph node without coming into contact with these cells based off of how it's organized. Because if you had an afferent vessel here and an efferent vessel here, you can imagine that it would be possible to just kind of like leave without encounter anything. But these lymph nodes very much are organized as filtration molecules or filtration centers. Can we see? Oops.
This slide is this is slightly out of order, but this should have showed us how we're draining into the lymph node in the first place.
And so basically you have this injury here, the sentinel cells. In this case, it's a pathogen carrying dendritic cells. Like Dr. DeLay was saying, the dendrites they sit inside of the peripheral tissue and they just innervate it. Their arms will stretch all over the place. And so they'll kind of act like little periscopes that are out there looking for it. And as soon as they can capture something, then they retract their arms and they walk through the lymph vessels to get to the lymph nodes where they can encounter the T cells and get the plasma cells to start producing antibodies.
And those could be other macrophages too. It's really just anybody who's capable of sensing that there's a pathogen and bringing chunks of that pathogen down to the lymph node where they can start educating the lymphocytes, B cells, T cells to do their specific job.
The spleen is also another, it's like the lymph nodes.
It's like a giant lymph node.
Basically. So there's different structures inside of the spleen. And so what you have is that there's these white pulp nodules that are scattered throughout the red pulp. And the red pulp is where old or damaged red blood cells, so this is basically like old erythrocytes, are removed from circulation. And then the white pulp is this secondary lymphoid tissue, which is like the lymph nodes. You have lymphocyte responses to blood-borne pathogens. And so you have the same sort of structure where you have this, a collection of B cells, you have a germinal center, and then you have access to the blood supply through which these mature cells are able to leave. And again, as in the previous diagram, this blue area is where you have the T cells that the B cells are then encountering.
Yep, don't worry. We're going to do all this again in more detail in just a minute.
This is a schematic of gut-associated lymphoid tissue. You have all of these secondary lymphoid tissues throughout the entire body. So you have lymph nodes that are scattered around your periphery that drain specific organs. You have the spleen, which is a specific organ that lives inside of the body. And then you have these patches that are actually on your intestines themselves. Like, for example, this is a really nice cross-section where you have this empty gut lumen. And then inside of the wall, you have this circular structure, which is gut-associated lymphoid tissue. It has within it dendritic cells, which are sentinel cells that will bring the pathogen to the B cells. The germinal center is where the sort of the maturation and the reproduction occurs, and then you pass through the T cell in order to be able to leave and get to the rest of the body.
Yep. So we're going to go into a little deeper into the antibodies now.
Right, because the B cells that are encountering the pathogen, their first line of defense is to select for the antibody that is specific to this pathogen. Because what happens when you encounter the pathogen is the fact that you have this zoo of B cells. One B cell is chosen that is specific to the pathogen.
Same for T cells. Yeah.
Same for T cells. And this is the structure of the antibody that is used to be specific to the cell. Basically, you have a heavy chain, which comes in 5 different formats, mu, gamma, alpha, delta, or epsilon. And this will depend basically on the structure, or this will inform the structure of the antibody. You have antibodies that are released just in this little Y shape. You have antibodies that are released in these floating clusters. You have antibodies that are released, that are not released at all. They're retained on the surface of the cell. And the type of antibody will be decided by the structure of this heavy chain. And what you can see here is that you have constant regions that are headed towards the surface of the cell. And then at the very, very tips, you have these variable regions. And the variable regions are made up out of one section of light chain and one section of heavy chain. The light and the heavy is very straightforward. Heavy is the big chain, light is the small chain.
So there's this really interesting thing that happens. I think I'll try to find a video for it really quickly too. But you do need to find the right cell. There's this clonal selection that has to happen inside of these secondary lymphoid tissues where you're trying out which B cell is going to do the job best. And
clonal selection is when you pick the right B cell and then you expand the population.
Right. And then there's this moment that happens also where you have after you find somebody that kind of does the job, what you're going to do is you're going to start really hypermutating that variable section. And so these cells go into this wild mutation phase where they're just trying out all these different combinations of those tips, those little antigen binding pockets. And they're going to do it and do it and do it until it matches perfectly. And as soon as they get a perfect match, they're going to, boom, proliferate, and you get tons of these B cells that are then going to go on and become plasma cells and be capable of secreting the perfect antibody to do the job.
And this kind of makes the entire process of the immune system make a little bit more sense. So what it is that you have themes and you have shapes that are recurrent in biology. And so the recombination of these antibody chains is creating the shapes that roughly approximate what will occur in nature. And then it's the secondary selection, this hypermutation that they undergo that really creates the super, super tight fit that gives you the kind of specificity that you need. The corollary of that is that if you have an inability to effectively mutate these ends, or the ends mutate in a way that is not favorable, you can have problems with the immune system. So the process of mutating the recombinant portion of this can be a place where the immune system can break. So this is just basically a chain model of how this works, where you have protein structures are often presented like this, where you're able to see the beta sheets in the alpha helices. And so what you can see is that there's this kind of hinge region here, which in the cartoon diagram is drawn as being very sort of stable. But in reality, it's kind of this floppy area which allows the antibody to kind of more effectively grab onto whatever it is searching for. So these pieces offer structural stability. Like I was saying, these don't change. They hold on to each other relatively tightly. And what you can see is it gives you this kind of globular structure in both places that is more for carrying these variable regions than it really is about doing anything for the immune system, except for the fact that this back end of it, so the back end of the constant region, is going to be recognized by the effector cells that are going to, or the phagocytes that are going to be swallowing these opsonized molecules or these neutralized toxins. These are the variable chains. We've done that.
Do you want to take a quick look at a video animation of this process?
Sure. What's the best way to do that?
I'll just stick a new slide in.
All right. So while he's doing that, basically what I was saying is that you have these different these different heavy chains. And the different heavy chains are going to be what is deciding what your antibody actually looks like. And so IgG is the antibody that you have that's secreted, and so it floats around in the blood. But that's not the only one that floats around in the blood. You also have IgM, which is actually a 5-membered ring of antibodies that are attached to one another through these constant regions down at the bottom. You have IgA, which is a dimer, which is capable of being secreted because it has this secretory component that links these two pieces together. And then you have IgD and IgE. And IgE and IgD are also secret.
Nice chart, actually, that follows on the next slide that makes sense.
Aha.
So, like, IgE is your main allergy response. So if you ever have like the sneezy hay fever thing, you're mostly secreting IgEs for those. These are called classes of Ig. The ones that like Dr. Benbury said we're mostly concerned with are the IgGs when it comes to COVID.
All right.
But yeah, let's take a step back and let's just look. I just found a cool animation of the entire process. This is just kind of a 30,000-foot view of encountering a pathogen, the cell actors we've talked about so far, and how they are going to work together to result in the production of an antibody. I think it's important for us to see that process all at once. So these are our macrophages. They could be dendritic cells. They have seen the virus, the antigen. They recognize the specific protein portion of it.
And they start displaying it on their surface.
Put it out there. They're holding it out.
So they're called antigen-presenting cells.
Now, there's just a T cell that's wandering around out in the environment that is capable of going around and checking out all of its compatriots.
It's surveilling. It's basically going around and it's touching all of the cells. So it basically walks up to all of these cells and it looks at their surface molecules and it's like, do you have an antigen in your surface molecule? And if you do have an antigen in your surface molecule, the T helper cell is capable of recognizing that and then initiating a cascade. So again, this is all like chemical signaling, right? So interleukin-1 is produced by the macrophage when it contacts the T cell when it senses the double whammy of a full antigen presenting structure, and interleukin-1 sends out interleukin-2, which is the secondary signaling molecule for this. And what this is going to do is now this is going to go to a different kind of group of cells. So you have two different types of cells, the cytotoxic T cell on the left and the B cell on the right.
Both of these are lymphocytes that are going to need to go through their maturation process.
And so what this is illustrating is that the cytotoxic T cells are basically, if you have a cell that is infected with a pathogen, whether it's bacterial or viral, what your regular cells are going to do is they're going to put pieces of that thing onto their surface. And when your cytotoxic T cell comes by and recognizes that that cell contains within it something that is abnormal, it is going to secrete chemicals that both damage the cell and induce the cell itself to basically commit programmatic suicide. And this is how you clear infected cells. B cell, on the other hand, are going to behave differently because they're not killing cells. And in this case, you have that when you come into contact with when a B cell comes into contact with antigen, that is when it starts to basically reproduce and to make more of itself. And so as you are producing more and more and more and more of these B cells, they start to secrete the antibody into the lymphatic fluid, into the bloodstream, and now this is traveling all over the body. And like we said, these antibodies are capable of binding to either toxins or pieces of bacteria. They get absorbed by these phagocytic cells. The phagocytic cells digest them and destroy them. And then you have these long-term B cells that persist. So basically, most of the B cells that you have that in this initial infection are going hog wild and replicating like crazy and producing as many cells as humanly or as many antibodies as humanly possible, most of those cells will die. It's very, very, very expensive for your body to continuously produce these because there is a lot of them. They are doing a ton of protein synthesis and they are using a tremendous amount of resources. And so it doesn't make sense to keep them around for a long time because it might be years before you encounter the pathogen again. And to deal with that, what you do is you have these memory B cells. And what the memory B cells do is that the memory B cells are capable of remembering and sticking around, and they persist sometimes for decades. And so when you have an immunization that works for decades and you don't need boosters, the reason that it works is because you have these memory B cells that are still around. They persist for long enough that if you ever encounter chickenpox or measles or whatever else, you will still have the ability to ramp your reaction up really, really fast. Okay, I'm not sure that there's anything. That just talks about everything that I said. All right. You have your B cell receptor, and the B cell receptor has this immunoglobulin that's on the surface, and it also has two little small immunoglobulin molecules that are necessary for being able to transduce the signal that is received on the surface into the interior of the cell. So this is a little dimer that basically is interacting with the constant region on the antibody. And when there's a shape change or a conformation change to the B cell receptor, it's transmitted into the cell through these alpha and beta immunoglobulins that are not technically part of the antibody complex themselves. The production of these antibodies is through this process that is called VDJ recombination.
Can we actually check out a different video about that?
For VDJ?
Mm-hmm. Well, for the bone marrow, or sorry, for the lymph nodes specifically.
Should we let's explain it first and then go to it? Yeah?
I think we did. Yeah, the last video wasn't as specific to the lymph node as I was hoping it would be. It was more of like a general pathogen chain.
All right, you got this?
Yeah. So slide 36. By the way, this animation was somebody's grad PhD project, which is kind of cool.
Okay, so this is, so before we go into it, this is basically what we're talking about here is affinity maturation. And so this is what Dr. Lee was talking about, where you have a B cell that's roughly capable of
Well, the affinity maturation is finding that perfect B cell. The hypermutation is the second part, which is the clonal expansion of the B cell that's okay into a bunch of B cells that are incredible. They have a perfect fit for that antigen. So this is a lymph node. We're going down deep into it.
So what you had here is that you had a B cell that is inside of the lymph node. It's being washed with all of this stuff that's coming from the periphery, and it binds the pathogen. It engulfs the pathogen, and then it presents it onto the surface. And so now it is an antigen-presenting cell, and it encounters a T cell. And the T cell is basically handshaking with it, and when it has done so, it basically is now activating the B cell like we saw in the previous video.
So it could have been any cell that presented it to the T cell too.
And this is where it's heading into the germinal center, where this is where they're really going to be activated. And
so it's moving into a particular location inside of the lymph node.
And so what's happening, this is a schematic of basically the fact that transcription is being activated where now its receptor is going to undergo a shift. And so what it's saying here is that the rate of mutation, so as it's transcribing the specific section, the rate of mutation goes up by a factor of 1 million. And go ahead.
Yeah, that's just wild. And it's only on this very specific part of the antibody is the mutation happening.
And so what's happening is that you have a receptor that is relatively able to contact the pathogen, and only a small section of it is undergoing this sort of crazy mutation. And so you're maintaining the rough ability of it. And so you develop all of these clones, and each clone has a different mutant of the receptor that appears to be working pretty well.
And they're interacting with the T cells, and the T cells are trying on that surface antibody, and they're like, does this fit? If it doesn't fit, they kill it off. The clone dies.
And so you have these dendritic cells that are inside of these germinal centers that are presenting antigen on their surface, and they're looking to see which one of the B cells is most effective at contacting and grabbing onto this antigen. And this is literally just a dissociation constant question, right? If you have the shape of your antibody is sufficiently strong to be able to grab onto the dendritic cell, then the dendritic cell is able to say that this one holds the strongest. And the key here is the fact that you have a limited quantity of this antigen that's available on the dendritic cell. And so these cells are literally competing to be able to grab onto these. And so the ones that are basically knocked off are viewed as being low affinity. And if they're low affinity, they can be destroyed because they're not particularly effective. And so the cell that's able to capture the greatest number of antigen-presenting compounds on the surface of the cell is going to be the one that is the winner of this competition. To the degree that it actually takes the antigen away from the antigen-presenting cell and then goes to the T cell, and then that
and it's like, let me live.
Right, because the ones that just pop off, they go apoptotic. They basically die. If you lose your contact with the dendritic cell, you're dead.
So you get this whole spread of affinity. Some of the antibodies work better than others.
Right, and so
this process just repeats itself and repeats itself and repeats itself.
And so what this is that you're seeing this reproduction of the cells. You have a population that's not particularly good at grabbing onto the antigen. It dies. You have the remaining population that then continues to mutate. It accumulates. Some portion of it dies. And then as you proceed through this exact same these set of steps over and over and over again, you end up with having an antibody that has incredibly high affinity, and you've destroyed all the other ones that don't have very high affinity. If you can differentiate into a plasma cell, you produce vast quantities of antibodies that are important for the immune response. And at this point, I think we're heading back into what we've already talked about, which is that you have these memory cells that stick around for a very long time.
Yep. And there's a similar process that happens in the thymus with the T cells, and we'll talk about that in a little bit. Cool.
The process by which you produce these antibody chains, the ones that you have in the first place before you go through affinity maturation, the receptor that just lives on the surface of the B cells, is through this gene recombination. So basically,
and this is happening in the bone marrow.
Mm-hmm.
So before you even get into the bloodstream, you need a diverse set of B cells, all with different antibodies on the surface that are more or less capable of talking with anything in the real world, any virus you might encounter. Maybe not perfectly, but more or less. They need to cover all the bases. And so this figure is kind of showing how that happens.
Where you have 4 different sections. So this right now is a schematic for just the heavy chain, but as you can see at the bottom of this picture, you have a heavy chain that is joined with a light chain and the light chain is matured in roughly the same sort of way, though it has a different process for the constant region. depending on what kind of antibody it is. And so basically what happens here is that you have several different variable domains. You have 26 different D domains, 6 J domains, and then a selection of C domains. And mutation of this, assembly of this is basically just different splicing mutants. And so there's a relatively random way in which this gets chewed up so that you have one LV domain, one D domain, one J domain, and then however many C domains are necessary to produce the constant chain that you want. But these are spliced up in different places, and so you basically get this kind of like word salad where you can take the beginning of one and the end of another, and then you end up getting something new that comes out of that by virtue of the fact that you had your starting set of options, and then you just slice up how you combine them. And because you have so many different possible combinations, your permutation of receptors goes up very, very, very quickly.
Because the real question is, how do you generate, I mean, think of all the combinations of antigens you might experience in the world. How do you, from a simple set of genes, how do you develop so much diversity? I was trying to find out exactly how many combinations are possible, but it's something extraordinary.
This is demonstrating
about 3 times 10 to the 11th combinations are possible. So through these simple recombinations, you're just recombining these simple segments.
So basically 3 times 10 to the 11th different versions of these endpoints, which once they are able to react to some shape, are then further matured through interaction, through basically dissociation constants inside the germinal centers.
And it makes sense because there's probably 3 times 10 to the 11th different little molecules that your body might encounter.
I don't know.
Approximately.
Maybe. I wonder about that. I wonder if there isn't like a smaller subset and you just like overproduce.
Maybe.
Right?
Why would you take up space with them in your bloodstream? It seems like you would have like lost them evolutionarily.
Yeah, that's maybe true. This is basically just a diagram that shows you that this happens both in the light chain and the heavy chain, and it's showing you how these things happen if it's in the genome or if it is in the RNA or if it's in the protein. So the endpoint for all of this is to get a protein, obviously, because antibodies are proteins. But you have splicing that is occurring on the level of the RNA, and you have recombination that is occurring on the level of DNA, which is how you basically siphon down to this final variable protein.
What's kind of interesting, this is all happening in the bone marrow, right? So this is a much earlier step than this business in the lymph node we were just talking about. This is literally how do you get those cells wandering around in the blood that are even capable of participating in that process in the lymph node. And so you do need to generate all this diversity, but you also need to make sure that these antibodies, which are at this point still stuck on the surface of the cell, they need to be stuck on the surface to do this job in the lymph node later. How do you get them to not stick to or react with your own body also? And so in addition to this, while you are having all this recombination going on, you're going to develop a lot of clones that are self-reactant in the bone marrow. And those are going to be killed immediately, because if they're not, then you're going to get all sorts of problems later, autoimmune disorders, issues where you basically have cells that are trying to attack yourself. And so there's a very specific process by which you are removing the cells that are overreactive. And they need to be in this real nice little Goldilocks zone of having this nice diversity, but not sticking to the self too much before they're even allowed to get involved in these other processes down the line.
This is just another schematic of the way that this works. This is, we're showing multiple different representations of this just because I think that it's super important to be able to visualize what this is and what is actually the sort of the effector molecule that is involved in the processes that we're talking about. But when we go through the COVID literature, it's rare to find something that goes deep into like this portion of the variable region on the antigen is responsible for, X and Y phenotype. In general, when we look through these papers, the obviously the antibodies are very, very important because they play a massive role in being able to actually produce immunity. But the level to which the papers discuss them is often not to this detail. Would you agree with that?
Yeah, and it's important too. I think we'll get into it hopefully when we start looking at the vaccines too, because the vaccines are essentially getting your body to produce these things, right? And the binding capacity largely has to do with that. The tips of these, there's these little fingers at the end here. These are called complementarity determining regions, CDRs. This is what are actually going to be sticky and bind exactly specifically. This is what's being hypermutated in that process in the lymph nodes we were talking about. But there's all this other stuff happening too, right? There's these different regions at which things could be going wrong. There's probably problems with, there's all sorts of opportunities for things to go sideways in the production of these antibodies that don't have anything to do with the complementarity determining region, especially concerning how flexible these hinges are, whether the antibodies are getting messed up and their shape is getting messed up in some other way that prevents them from binding to the endpoint.
But if you have fundamental problems with antibody production, you are very, very unhealthy to begin with. Like, you cannot live in the world and be encountering anything from bacteria to other people if you don't have a pretty functional antibody system. And so when we start talking about immunocompromised patients with either overactive or underactive immunity, there's a degree to which these segments of the immune system can be broken, but that already creates a starting point of pretty severe dysfunction that, can go anything from having to be fully isolated all of the time because you don't really have an immune system to having an immune system that is chronically attacking your own cells because the function by which it can stop that is broken.
Yeah, your body can actually make antibodies to antibodies too. So there's lots of levels at which this can go wrong.
It's antibodies all the way down. Schematic, basically, that you have this common lymphoid precursor, you have the B lineage that is in the bone marrow, they are matured here, they go into the blood and lymph. Inside of the lymph nodes, and inside of the spleen, they encounter pathogens, they become fully matured, then they're either plasma cells or they're memory cells. This is a very cool chart. So primary response and secondary response. Do you know?
Yeah, I'm not sure which book this is from, but essentially this is just showing you that once you've essentially made these B cells in the first place, they're going to they're going to do their job much better the next time around. And this is the cool process about this idea of memory. This is why it's called the adaptive immune system in general, I believe, is because you're seeing something, you're having a reaction to it, but in the future, and this is the principle behind vaccines in general, is that once you've seen something, it's not going to be a problem in the future. This obviously hasn't played out perfectly with something like COVID and respiratory viruses in general because there's lots of reasons, but certainly they're mutating very quickly. They're spread by the air, which allows them the opportunity to live in a lot of different hosts and also increases their mutation rate. But with a lot of diseases, you may recall you get vaccinated as a child, say against smallpox or something like that. You'll never ever need to do it again because you'll still have those antibody responses years and years and years later.
I think nobody since the 1980s has been vaccinated against smallpox. You and I might not be the only ones in the room.
Oh, all right. Well, how about measles or something like that?
Chickenpox, yeah.
Chickenpox. Yeah, chickenpox is a good one if you've ever had it. I actually had chickenpox, but I think most of the kids get vaccinated against it these days.
I think this is the portion of the class where we just list diseases that we've had. So basically, the key to see here is the fact that when you have not seen a pathogen, you have a lag phase. And the lag phase is because the only B cells that you have, the only cells that you have that are specific to the pathogen that you are encountering, have not yet been differentiated. They have been differentiated to the degree that they're not going to attack you, but they are not necessarily able to start producing antibodies in huge quantities. Plasma cells are a massive part of the adaptive immune response because they are responsible for opsinization and neutralization because of all of these secreted antibodies. If you don't have mature B cells, you can't start producing that yet, because the worst thing to do would be to start creating massive quantities of antibodies that are not specific to the pathogen, and they get in the way of the antibodies that you produce down the line that actually are specific to the pathogen. Because all of this this is all about receptor occupancy and the ability to actually get into the spot where you need to be. And so if you have something that's kind of like just an okay antibody and it's circulating in large quantities because your naive B cell has turned into a plasma cell and has started secreting it, what's going to happen is that, let's say that you actually are capable of coating a virion in antibody. Well, you have to actually coat it well enough to be able to hold on to it, to drag it, right? Because what happens is that your phagocytic cell is going to come up and it's going to grab the back end of those antibodies, and then it's going to start to pull everything in. And if when it starts to pull, it releases, then you're screwed. There's no real other way to put it. And so you have to tune this tightly, because if you just start producing whatever works in this first moment, you're going to end up creating a lot of problems down the road.
One thing I wanted to add here is you'll see this terminology a few times. The IgM versus IgG, this is the class switching. I guess we had these slides a little bit out of order. Sorry about that. But the IgG is that secreted final form. These are the antibodies that they're dumping into the blood. If you hear about people getting monoclonal antibody treatment, it's the same idea. Although sometimes those are like slightly modified versions of IgGs. But the IgM is the surface receptor. These are the antibodies that are stuck on the outside of the cell when it first comes out of the bone marrow. It's called.
You can remember this IgM as Ig membrane.
Yeah, great idea. Nice. And so not until it becomes a plasma cell are you seeing, and that's why the little second hump here is a bit later than the first one, because once the cells have actually gone from, okay, we have an antigen that we're dealing with, they're going to expand, and you're going to get all the pools of the ones that are sort of doing the right job are going to appear, but not until later, after we have that whole hypermutation phase, do we get this secretory form of the antibody. However, later, that one's going to know those plasma cells are going to go straight into action, and you're going to get a huge bolus of that secreted antibody during the second exposure, which is why you hardly ever get sick, because your body's just tagged and removed the pathogen the second time around. And honestly, if the vaccine is 100% effective, this is exactly what happens as well.
But respiratory viruses are hard to get 100% effective vaccines for because you have a lot of mutation. And so if you have, this is kind of where things start to get really screwy, is because if you start with this population of activated B cells, and so you have IgG that is being secreted to a specific antigen, and the antigen is the shape of the virion. If the virion is capable of changing that shape, then this secondary activation either will not happen, or if it does happen, it will be insufficiently specific to allow opsinization and neutralization to occur. And so this is the place where you really start to see the effect of mutants and the effect of escape variants, because it subverts the ability of the immune system to do this, which means that what you start out is you go back to this very, very starting point where what you're having is the primary exposure as opposed to a secondary exposure. Okay, very quickly. So basically we have these lineages of different cells. And so we've been talking about the lymphoid progenitor lineage, which has to do with the B lymphocytes and the T lymphocytes. The fully matured B lymphocytes are called these plasma cells. The plasma cells are the ones that are circulating in the bloodstream that are able to, and the lymph fluid, able to produce massive quantities of antibodies. The plasma cells, once the active infection is over and done with, these will die off. Only the memory cells will remain. T lymphocytes, there's multiple different types of T lymphocytes. which we can kind of crop into a little bit because honestly, the world of how many different T cells there are is pretty big.
Yeah, I think we can have some charts in a second. But the key thing to remember here is that the development for everybody is going to start, the precursors are going to be developed in the bone marrow. Very shortly after development, however, the T progenitors are going to go to the thymus to develop. Whereas most of the maturation for the B cells is happening in these secondary lymphoid organs. I guess some of this recombination is happening in the bone marrow to some extent as well. But a lot of that's happening, all that diversity of receptors is happening both in the bone marrow and in the lymph nodes. With the T cells, it's happening in the thymus. And so most of you probably don't even know where your thymus is, but it's a little glandular organ that sort of sits like somewhere around here. And it's going to go, the cells there are going to go through a series of recombinations and positive and negative selections where they're tested against, again, they don't want to react with the cell. But you're, again, trying to develop diversity. This time, the T cell has its own receptors. So unlike that surface antibody, there's something called the T cell receptor. And we're going to go into that.
But let's take a 5. So we'll see you back at 6:10.
Again, those T cell receptors are generating a great deal of diversity in a very similar process to the recombinatory the recombinatory programs that we talked about with B cells. And you're selecting for, not only do you want a diverse set, but you want cells that are capable, in this case, of binding the cell through this MHC receptor, but not too tightly. And there's just these perfect cells that end up being squeezed out of the thymus eventually.
What I love the most about this video is how sterile and empty the interior of the body is presented, where it's like you're walking down this massive empty hallway and there's just absolute peace and silence. And there's 3 cells in the entire body. Obviously, this is a very crowded process. They're all banging up against each other. And this was illustrated in a childhood thymus because this happens early. This is basically something that you are set up with when you're a child. And one of the reasons that COVID is so dangerous for people as they get older is that as you age, your thymus begins to shrink. And so it becomes less capable of maturing new populations of T cells. And so your T cells that are still around might have been around for a really long time, and they're getting tired and they can get exhausted. And if they're not capable of doing their jobs, which has to do with activating B cells and with killing cells that are infected, then you run the risk of ending up in a situation where your immune system isn't functioning the way that it's supposed to because you can't clear the cells that you want to clear.
Along those same lines, this is a huge part of the reason why we see leukemias in children, because you have a tremendous amount of cell proliferation in the early years of your life, where all of these white blood cells, right, the whole immune system is essentially being constituted early in life, and you have all these hyper rearrangements. The cells are dividing like gangbusters, and so you have the opportunity for things to go wrong. Of course, there's different genetic predispositions to that. But sometimes things just go sideways, and you get a runaway clone. One of these cells escapes, and it's not supposed to get out of the thymus, or one of the cells escapes in the bone marrow. It's not supposed to. It's supposed to die, but it didn't. And you can get all sorts of interesting and terrible consequences as a result of that, from autoimmune disorders to leukemias.
We're giving a pretty, like Dr. DeLay said at the beginning, this is a very 30,000-foot view. We're not going into the sort of the intricacy of the receptors that you need to know. We're touching upon some of the signaling molecules. Like they talked about chemokines. We haven't talked about those. We haven't talked about the sort of the physical movement of the cells, because we will talk about those as they become relevant for the papers that we look at. There's just, you can take an entire year of immunology and not exhaust what it is that you need to know. And so what you're getting from us is you're getting a very sort of general picture that is sufficient for us to be able to discuss COVID and its biology. rather than focusing on the nitty-gritty of like what receptor does what. If you go into grad school, if you go into medicine, you will have to know this stuff. And so our goal is to present you with a baseline so that the next time that you see it, you remember a little bit more, because memory in the brain works similar to memory in the B cells, which is that when you've seen something for the very first time, you very much are like the naive B cell that's just like, what is this?
Yeah, normally when we teach immunology, we have a whole semester to just do, we'll spend several lectures on T cell maturation and development, several lectures on B cell. But we want to make sure that we can cover all of these at least once, so that when we get into the details of it in the papers, it will at least be familiar to you. So I think these are some salient points about the T cell system that are going to be helpful. Especially, we're going to see this word CD8 and CD4 a few times, which I think is worth remembering in the literature. In that video, actually, we just watched, there was a lot of talk about the CD4 and CD4. At some point, they're double positive. They say the cells are double positive. They express both of these. So what is a CD marker? Well, a CD is just a particular labeling schematic that the scientists have come up with for what are the names of these proteins that are sticking off of the surface of these lymphocytes, the T cells, or any cells. B cells have CD markers also. The word CD stands for cluster of differentiation, I believe. And these are just specific proteins that act as lock-in keys when the cells are moving around the body and going about doing their work. So CD4 positive cells, these are the ones that express CD4 versus the cells that express CD8, have a different job in the body. They're called helper T cells. They're largely there for orchestrating the processes around the antibody production we're talking about out in the lymph nodes. I believe that there was a T cell in the first video that was out there doing some work in the field. The CD8 cells are often called killer T cells. They're actually the ones that are going around there and finding the cells that have been infected, say with a virus. So all of the cells, like Dr. Benneberry mentioned at the beginning of class, when you get infected with something, your body is going to start presenting little pieces of that virion, little pieces of the viral capsule on its surface.
And this is these MHC molecules. So we've talked a lot about this, where it's like you have something inside of you and you end up putting it on the surface. You become an antigen-presenting cell. You present antigen through these MHC, major histocompatibility complexes.
And so the CD8 cells, their job is essentially to go around tasting all of these little platters, these MHC platters, and seeing what your cells are holding out. And normally, if your cell's healthy, it's just holding out a dish of itself. And because the cell has been matured so carefully inside of the thymus, it knows not to flip out when it sees a little piece of itself, and it moves on to the next cell. But every once in a while, it's going to encounter something that isn't itself, which is going to lead to it initiating this whole cascade. And since they are called killer T cells, it's going to kill that cell, because you don't want a cell that is making stuff that isn't it.
And this is a theme that we're coming back to over and over again, right? This is the process of maturation of these learned, educated immune cells is very important, because dysfunction is catastrophic for the body. There's these 4 different classes of T cells: cytotoxic, helper, memory, and regulatory, which we will talk as we go what they do and how they're important. The CD receptors differentiate them, and they behave accordingly. This is a sort of physical or a drawing recapitulation of what we've done, which is that mature T cells lose either one or the other of the CD receptors. They maintain their T cell receptor, though. Like, your T cell receptor is something that every single T cell is going to have. And that is very, very important. You're on the headphones.
Yeah, the T cell receptor is what's interacting with this platter molecule I described, the MHC molecule. I want to make sure we leave enough time for questions to in
So
this?
let's do 15 minutes of questions at the end. So we've got a little bit more time to get through it.
Okay, I think this goes to 6:50.
6:55?
I think is it? I think it's just
6:55.
6:55. We started a little late.
We did. Okay. So basically, T cell receptor is here. It has a different shape from the B cell receptor. The B cell receptor is this Y-shaped receptor. This is not a Y-shaped receptor. It's straight up and down. CD3 is the companion molecule that helps with signal transduction. So the same way that the B cell receptor, its transmembrane domains are short, which means that it's anchored in the membrane, but it doesn't actually have anything dangling down to the interior of the cell to be able to create the signal transduction cascade that's necessary for any kind of genetic changes. And so you have accessory proteins, which are CD3s, that are part of the T cell receptor complex and are necessary for being able to actually do the job of the T cell. There are two classes of T cell receptors, and their structure is roughly the same. We're going to get into this when we're actually in the literature, because sometimes you'll have different classes that we talk about. And so basically make a note of this in your heads right now. There's two different types of T cell receptor. They both bind antigen. They both have a transmembrane region, a little bit of a cytoplasmic tail. They have two chains. Beyond that, we'll get into the differences of them once it's actually relevant. I'm not sure that it's worth going into right now.
No, this is just too much information.
All right. This again.
This is more schematization of the same video.
Do we have the MHC receptors in here today?
That's coming next.
I'm very excited for that, because the MHC is a really, really fascinating piece. This is
kind of illustrating the, at least on the right side, this is what I was talking about, the cells going around and surveying. So an antigen-presenting cell is just any cell in your body that's holding out a piece of either itself or a piece of an infectious molecule. And similarly, other molecules can go out and find the bad guys in the field and bring little pieces of them to the CD4 cells on the left, who are going to go out and do their job and orchestrate the attack.
The difference between the mature helper cell and the cytotoxic cell is the fact that they both secrete stuff, right? The mature helper cell is secreting something that helps mature the B cells, helps activate the rest of the immune system. On the other hand, the cytotoxic cell is secreting molecules that are causing apoptosis, that are causing programmed cell death. And so they end up having very, very different functions because of that. You cannot function without either one of these. So you have to have CD4s, you have to have CD8s. They balance. There's also regulatory T cells, which really help activate the inflammatory nature of the immune system, because any one of these cells is really capable of starting off a cascade that's going to cause a lot of problems down the road. And so you create a third kind of T cell that's basically there to be like, hey, we seem to be getting too inflammatory and I'm not sure that this is a good idea. And it can tamp things down by either affecting the maturation of them or affecting the reactivity of them. Or if it seems to think that things need to be much more active and hotter than they are right now, it can kick it over into that direction as well. But before we get to the question segment, I really want to do the MHC, which is the major histocompatibility complex.
This is the platter that's presenting either the bad guys or yourself to the T cell system.
And the MHC is kind of an astonishing thing because it is a mechanism for being able to identify self and other. And on this slide we have three different types of cells. We have target cells, I guess 4 cells, sorry. So we have target cells up here on the left. We have CD8 T cells down here on the left. We have antigen-presenting cells, which are up here on the right. CD4 T cells down here on the right. So the difference between the reason that we have target cell versus antigen-presenting cell is because when you interact with the CD8 T cell, you become the target of the CD8 cell. You are its prey, basically. It will kill you.
Unless yourself.
unless you're
healthy.
yeah. And then the CD4 cell, it's interacting with an antigen-presenting cell, which is basically every single other cell.
Well, like we talked about right at the beginning of the lecture, we're like, well, there's these sentinel cells, like the dendritic cells, or it could be a macrophage. Somebody who's like eaten maybe a little piece of something bad and is like, hey, this is over here, check it out.
But all cells have them, right?
All cells do, yeah. But some of them are sort of like specifically tailored to this, right? And so you don't want to kill the macrophage off just because it's eaten a bad guy. And so it's going to maybe activate a CD4 cell. And these are through different MHC receptors, class 1 and class 2.
And so HLA is also a different name for this MHC. So MHC or HLA,
there's always two names for stuff in immunology, at least. So yeah, especially you med students get used to it. It's going to be kind of a fun ride.
So the difference between these two types of receptors, MHC class 1 and MHC class 2, has to do with a couple of things. Number one is that you have a different structure of the receptor. So MHC class 1 has only one transmembrane domain. MHC class 2 has two transmembrane domains. And the way that it interacts with the CD8 cell is different as well. You have this T cell receptor that is consistent between both of them. So CD8 has the T cell receptor, CD4 has the T cell receptor. The antigen is in the same position, but the difference is the fact that your CD8 molecule will stabilize at a different place on the MHC. And so it's basically lock and key in this way, where after you've bound this antigen presenting pocket, if you don't have a place on the surface for your CD8 to bind, this interaction won't actually happen. And so you'll have to have the CD4 molecule in order to basically hug the entire structure to stabilize it, to help the interaction actually happen. And what's also different about this is the way in which this antigen-presenting cell packages the antigen, because there are two different types of antigen packaging pathways. One is you are packaging stuff that is actually inside of the cell, and the other one, you are packaging things that have been digested by the cell. And these end up having very, very different reactions when it comes to interactions with CD8 versus CD4, right? Because if you have something that is inside your trash can and you pull it out and you're like, hey, I've disarmed this and I'm going to put it on my surface, and then you have one of these T cells come by, you really don't want to encounter a cell that's going to kill you after you've already neutralized something, because you've dealt with the problem. You're not infected, you're not under threat, you're basically just like, hey, check this out, I found this, you might want to know about it. And that's why you want to interact only with the CD4 cell. But if you have something that's actually inside of your cytoplasm that is harmful or damaging or dangerous, then you want to be encountering a CD8 cell, because that's going to be the cell that's able to say, hey, this cell is really sick and we need to kill it because it's sick and dangerous, and if we allow it to keep surviving, then it's going to start causing more problems because it contains either a bacterium or it contains a virion or whatever.
Yeah, if you want to go here, skip that one. Come on. We have a nice illustration of what you were just talking about. Go down. There you go.
This one's hidden for some reason.
This guy right here.
This guy?
Yep.
Yes, exactly. So basically, this is going to be the breakdown of something that's inside of the cell. It gets packaged into the MHC molecule, and then the T cell receptor is able to bind it. Do we have the...
I love this picture. There's another way of thinking about it. So, you have this infectious chicken that makes it inside of your cell. This could be a virus or whatever. It could be a virion or a bacteria. Gets chopped up by the proteasome into all the composite pieces into a little protein. And then the HLAs are putting them out. This is where I got this platter idea, by the way. And that guy up there, by the way, CTL is another word for these CD8 killer T cells. This is the watchdog who's going around with his T cell receptors and tasting all these platters. The more ways you can see this, I think the more it will begin to stick, and it will make more sense when we start talking about, especially in terms of the symptoms, because a lot of what's happening in COVID in terms of whether a patient is crashing or not has to do with whether these different processes are working out as they're designed to do. I don't know if designed is the right word, but as they have been developed optimally.
This actually wasn't my favorite.
That video?
No, oh, this is the one that I want. We'll go back to the video in a second. So this is the graphic that I was really looking for, which is we have the MHC class 2 versus MHC class 1. So if we go back and we think about the way that which MHC molecule interacts with which T cell receptor, MHC class 2 is going to interact with your CD4 cells, which means that it's not going to be killing the cells. It's going to be activating the immune system. MHC class 1, on the other hand, is going to be interacting with your killer T cells. And the pathway by which these are packaged is exactly reflective of the risk. So you have an extracellular antigen that's been taken apart that gets packaged into MHC class 2. So you have it's been defanged, and now it can be interacting with these helper T cells. But when you have something that's actually an intracellular antigen, which is that it's living inside the cell, that's when you call the alarm bells. And I view this as being one of the most elegant mechanisms inside the body for being able to differentiate necessary functions. The structures aren't that different. The final behavior is very different. And all of that is based on the pathways of packaging and the pathways of receptor ligand interactions. And it just kind of epitomizes for me the mastery of the immune system when it comes to actually being able to deal with threats, where it's like it's very finely tuned to be able to know what is worth reacting to and what is not worth reacting to.
I think we should finish up this lecture with slide 68 here, which is kind of an overview of all of the different T cell roles, because ultimately some of these terminologies are going to keep coming up over and over again, especially this idea of becoming an effector. What is that? And so you can see here that it's actually fairly simple. The T cells are generally making contact with other cells, and they're going to do more or less one of three things. They're going to kill off the cell that's infected. They're going to activate other cells to get involved in the combat, or they're going to help B cells develop. And I think this is a really good illustration of those three separate pathways, and we're going to keep coming back to them. Again, we're going to put this lecture online. You can come back and pause this section. I would recommend it if some of these things didn't make sense, hopefully after we talk about them. And we should take some questions now too, so that we can make sure that something didn't get glossed over. But that's it in a nutshell. Those are your two main adaptive cellular systems, the B cells, the T cells, and then you have all of these very rudimentary scrubbers, little foot soldiers called the innate immune system, which we talked about last time. And we should be ready next time to talk about viruses, and then we're going to put it all together. We're going to put it all together and do COVID.